female Anopheles quadrimaculatus is a recognized malaria vector in the south. Various types of malaria occur in this region. How well does this mosquito transmit these different malarias? How well will it transmit foreign malarias introduced by infected persons? How will our different malarias infect other species of mosquitoes, whether native or introduced? To answer these and related questions, a method of laboratory testing has been developed. The answers to these questions are essential for appraising the danger of importing malaria carriers and for applying proper control measures if they are necessary. The United States Public Health Service established a program to determine whether native anophelines can be infected by and transmit the foreign parasites brought into this country. These studies must be conducted with humans, since the malarial parasites which affect man do not live in lower animals. Parasites which will infect mosquitoes are obtained from patients who have primary or chronic infections of malaria acquired in foreign countries. Adequate supplies of the Anopheles species to be tested are reared on a large scale in carefully managed insectaries. Susceptibility of these mosquitoes to foreign malarias is determined by dissecting the insects at intervals after they have fed upon a person infected with one of the imported malaria parasites. Testing the ability of the species to transmit the infection to susceptible humans is more difficult, and few individuals are willing to expose themselves to experimental malaria. However, malarial infections have recognized therapeutic value in treatment of cerebrospinal syphilis. Mosquitoes infected with malaria are allowed to feed on such patients. With facilities for supplying the three prerequisites: 1. Infectious parasites. 2. Infectable mosquitoes. And 3. Susceptible humans. Tests can be made of the ability of native Anopheles to become infected with and transmit foreign malarias. The first step is to obtain a blood film from a patient known to have the malaria selected for testing. This film is stained in the usual manner with an ordinary blood stain, and then examined for gametocytes. When examination reveals gametocytes, infectivity and transmission tests may be initiated. Mosquitoes to be used in the experiment are transferred from the emergence cage to small feeding jars. From 1 to 5 mosquitoes are placed in each jar. Ends of the jars are covered with bobbinet through which the mosquitoes can bite easily. These mosquitoes have not had a blood meal, but have been fed on a dextrose solution. They should bite and take blood readily. Moist chambers, a thermometer, and materials for making blood smears are packed. Jars containing mosquitoes are placed carefully in a safe container. In the ward, feeding jars are placed along the sides of the patient's legs. Several hundred mosquitoes may be fed in this manner on a single patient during a period of from 15 to 40 minutes. When the mosquitoes feed, the gametocytes present in the patient's blood are drawn into the mosquito stomach, and the cycle of plasmodial development in the insects begins. Note the drop of fluid colored by the presence of red cells being forced from the anal opening as the mosquito becomes fully engorged with blood. While the mosquitoes are feeding, additional blood smears are taken. This procedure is to determine the number of infectable parasites present in the patient's blood and if viable gametes will develop. Exflagellation of the male gametocyte is encouraged by breathing on the slide. The slides remain in a moist chamber for a 5, 10, 15, or 20-minute period. While the smears remain in the moist chamber and the mosquitoes are feeding, the technician records pertinent information concerning the patient's case history. At intervals of 5 minutes, a slide is removed from the moist chamber. Excess moisture is wiped from the back, and the slide marked to indicate the length of time which it remained in the chamber. The slide is then placed in a slide box and allowed to dry. Subsequently, the slide will be stained and examined for the formation of male gametes. The room temperature at the time is recorded since the rate of exflagellation is affected by temperature. The mosquitoes have now been allowed ample time for feeding. When the mosquitoes have engorged, the feeding jars are removed from the patient and packed carefully for return to the insectary. These mosquitoes are now potential vectors of malaria. Escape of even one of them could be dangerous. Care in handling them is imperative. In the insectary, these mosquitoes must be cared for under controlled conditions for the 10 days or longer required for plasmodium development in the mosquito. Count is made of the engorged mosquitoes. Those which did not feed are destroyed. The jars already labeled with the patient's name are now marked with the date of feeding. After the feeding jars are checked, they're placed in trays where they remain until the mosquitoes are killed for dissection or are fed on a non-infected person. Daily, the mosquitoes are fed with a dextrose solution. In the laboratory, the technician stains and examines the blood films taken from the patient at the time of feeding. By studying the slides, information is obtained concerning the infective stages of the parasites. Gametocyte density is determined, and search is made for exflagellation of male gametocytes. Exflagellation indicates that viable gametocytes are present, and the mosquito will probably become infected. At intervals of 2 days, starting with the 6th day after the infective blood meal, sample mosquitoes are dissected to determine the rate of infection. The mosquitoes may be killed by a cloud of tobacco smoke blown into the feeding jar. Dissection is done under a binocular dissecting microscope. The mosquito is placed on a slide and the legs and wings removed. The body of the mosquito is transferred to a drop of saline and held under gentle pressure with a broad, flat needle. The head is severed and the salivary glands are extracted with a sharp dissecting needle. The salivary glands are placed in a drop of saline for examination. The body is held still with a broad, flat needle and the stomach removed by grasping the terminal segment with a pair of sharply pointed tweezers or with a needle and drawing out the abdominal contents. The stomach is also placed in a drop of saline for examination. A cover slip is placed over the dissected stomach and glands and excess moisture removed with a bit of cotton. The slide is then examined with a compound microscope. If oocysts are present in the stomach wall, they may be seen with a low-power objective. Oocysts are small round bodies resting between the outer membrane of the mosquito stomach and the inner epithelial cells. The number of oocysts present indicates the intensity of infection. Oocysts are visible in about a week after the blood meal and reach maturity in about 2 weeks. When the oocyst is mature, it ruptures, liberating sporozoites into the body cavity. This photomicrograph shows the liberated sporozoites and illustrates their motility. The salivary glands are most satisfactorily examined after crushing by applying gentle pressure to the cover slip. Sporozoites, if present, are released. Sporozoites, which may be seen in the salivary glands after about 2 weeks, indicate that the malaria infection has matured in the mosquito. This picture was made from a stained slide. To test the ability of the mosquito to transmit malaria, mosquitoes from the lot tested are allowed to feed on a non-infected person. The mosquitoes are then killed and dissected. The actual number of infected mosquitoes is determined. Each step in the experiment is recorded in detail. Approximately 2 weeks later, if transmission was affected, the person will develop clinical malaria. and parasites can be demonstrated in his blood. This is conclusive proof that the native vector is capable of transmitting the malaria. We have seen the 6 important steps in testing for transmission of malarias. 1. Blood from the patient with foreign malaria is checked for presence of gametocytes to be sure that forms of the parasite which can infect mosquitoes are present. 2. Anopheline females are allowed to feed on a malaria-infected person. 3. These mosquitoes are cared for during the 10 to 14 days while the development of parasites take place, and are checked periodically to determine rate of development. 4. The infected mosquitoes are fed on non-infected persons to determine if transmission occurs. 5. The mosquitoes are killed, dissected, and checked to determine the percentage of infection. 6. The patient is followed daily, both clinically and by blood smears, to detect the development of malaria infection. This is a procedure for testing the ability of native anopheles to acquire and transmit malarias. The results obtained assist in appraising the public health significance of native malarias as well as of foreign malarias which are introduced.